Thank you. Let me share my screen. Can people see my screen?
Yes.
Wonderful. And let me do full screen. All right. And first of all, I would like to very much thanks Bob Wright and the other organizer for inviting me, and a great pleasure to share with you some of the work we have done and using the Harvard Superfund data. And in particular, I'm going to discuss how the misspecification of the environmental effect will impact when we study the gene-environment interactions. So this work is led by my former student, Ran Sun, who's currently a assistant professor at MD Anderson Cancer Institute. So, let's talk about the gene-environment interaction. So as we know that in the past decade, there have been an increasing number of the genome-wide environment interaction studies. But the number of replicable GE interaction is only a handful. And so therefore, how can we enhance the reproducibility and the replicability of the gene and environment studies? So then a natural question to ask is, if we incorrectly specify the exposure effect, how will that impact our gene and environment interaction studies? And then the answer will be yes. So for example, if you look at the following QQ plot published in Emily's 2014 paper, and then you can see that if one look at the main effect, that basically is this black curve, and it looks very good. But when one add in the gene environment interaction, you can see that become off. And so that tells us that if we misspecify the environmental exposure effect, that could impact the GE interaction effect. And we know generally when we study the environmental effect, it can be complicated. So let's me introduce the Harvard Superfund. And so this basically motivated this investigation. And in the Harvard Superfund, this is early Superfund study, not the current cycle. And so the goal is to, one of the goal is to study the gene environment interaction on in the effect of toxic metals and on the infant neurodevelopment. And so for this particular project, we focus on two outcomes. One is a mental development index, the MDI. The other is psychomotor development index, the DPI. And for illustration purpose, I'm going to focus on a single exposure, that is lead concentration collected using the cord blood. And so in this study, we did the GWAS studies, and then we genotype about 550,000 SNPs. And then in this study, and there are multiple sites, and I'm going to focus on the Mexico side and also Bangladesh side. We also have the Tarquit side. And for the Mexico side, we have about 400 infants. And for the Bangladesh side, we have about 500 infants. So we started from the standard approach. And so basically start from a linear or logistic regression models, and then include the main effect of the G, environment effect of E, and also GE interaction, and also several covariates. So a major challenge is how can we model the lead effect? And then how the lead effect modeling could impact the GE interaction. So for the SNP, we just analyzed one SNP at a time in the GE interaction models, and also for the lead, and then we include both the mean effect and also the interaction between the individual SNP and also the lead, and also other confounders. And so then this raises the question, how can we model the lead effect? And so we should use the lead in the drinking water, and or in the daily exposure to lead, or the blood concentration of lead, or should we take a log transformation of the lead concentration, or we should create a categorical variable for lead, or we should use the lead in diet. So there are many different choices and to modeling the lead effect, and how are the different choices going to impact the GE interaction. So let me introduce the standard GE interaction models. So here you can see that we have the SNP here and the lead here, and the SNP by lead interaction, and also other covariates, and so such as age, gender, and so on. Our interest here is to study the GE interaction, basically testing the beta GE equal to 0 versus beta GE not equal to 0. So we analyze one SNP at a time, and we analyze the Bangladesh data and the Mexico data separately, and then we perform the meta-analysis. So let's focus on the standard approach, and this is captured by this red QQ plot, and this is for the beta GE, the interaction term. So if you look at on the right, that is the QQ plot of the interaction, GE interaction, and using the Mexico data. And so if you look at this red curve, this is the standard approach, and by modeling the lead effect linearly. And then you can see this depart from the 45-degree line very early on. And the Bangladesh data looks better. And now when we did the meta-analysis, and so you can see that is the red curve here. And so you can see the Bangladesh data and also the Mexico data after doing the meta-analysis, and then the QQ plot is off, is below the 45-degree line. And so we were puzzled, what went wrong? So maybe we thought, oh, maybe we did not model the SNP effect right. But this is a QQ plot. If the mean effect, you can see it lies on the beautiful 45-degree line. So that indicates we model the population structure okay. And then something else went wrong. And so when we did the further digging, and we found out it probably the lead effect is misspecified. It probably is not a linear effect. And so after then when we test, as I mentioned, when we look at the QQ plot of the main genotype effect, Q, it looks pretty good. It's on the 45-degree line. And then we found that the exposure effect, the lead effect, it does not seem to be linear. So this reached a natural question: how misspecification of the environmental effect and the confounder could impact the inference on the gene and environment interaction. So we did the analytic analysis, and also we did the simulation studies. And so here, for illustration purpose, I'm going to only focus on the analytic result. The simulation result support this calculation as well, and also it has very similar result. Okay, so here the model we consider is the main effect of the G and the environmental effect, and also GE interaction. This beta GE is a major parameter of interest, and also we have a bunch of confounders. And we consider multiple models, and so suppose this is the model we fit. The true model is listed on the left. You can see that in the first model, the true exposure effect is the E square. But then when we fit the model, we misspecify the exposure effect as a linear effect. And then here, we want to see how that impacts the beta of the interaction term. So on the x-axis, that indicates a correlation of, sorry, can I continue? Okay, all right, so the x-axis, I'm not sure why I hear the echo. Can you hear the echo too?
It's okay now.
Okay, let me start again. Okay, now it's okay. Okay, so the x-axis is a correlation between the red term. So in the first example, that basically we assume GE are correlated. And so then the y-axis is the bias of the interaction terms. So as you can see in the first example, we basically misspecify the environmental effect because we want to look, then we fit the interaction models. And so this is captured by this blue curve. So as you can see that when the G and E's are independent, and then we don't see the bias. But when the G and E are correlated, and then when we misspecify the exposure effect, we see this bias curve, this blue curve. The second example is when the exposure, sorry, is a typo here. When the G and the confounders are correlated, this is the red curve. G and the confounder are correlated. So when we misspecify the exposure effect, and so the true exposure effect is E squared, but we misspecified as a linear effect. And so this is captured by this red curve. Then you can see that the interaction term is biased as well, unless the correlation 0. And the third example is, suppose I don't know what the true exposure effect. Suppose the true exposure effect should be the true light level at using the blood, but we use the light level using the water. And so the true exposure effect should be the M, that is the exposure effect in the blood, but we misspecify it using the light in the water. And so if the exposure and the genes are correlated, and then you can see that this will be the green curve. And so that means that the estimate of the interaction term will be biased, and the bias becomes stronger when the correlation between the G and the exposure and the misspecified exposure becomes stronger. But if the correlation 0, and the bias is okay. And the last example is the true effect is also the E squared, but we introduce this additional non-confounders, and then this also causes a bias. That is a purple term. That's a purple curve. Unless the correlation between the G and also the confounder, that is the water light level, is 0. So the summarize the finding here, what they tell us is, when the exposure effect is misspecified, and then the interaction term, G interaction term, is biased if the G and the E's are correlated, or if the G and the confounders are correlated, or the confounder is misspecified. And then when the correlation between the G and the E's are 0, basically G and E are independent, or G and the confounders are independent, and then the point estimator of the interaction term will be unbiased, basically this point here. And so what that means is the point estimator is okay when the G and E's are independent. And the natural question is the testing still okay? Even the point estimator is okay, but whether the test is okay. So this is the result. So suppose we assume that the G and E's are independent. When G's are not independent, we know that the inference for testing for the GE interaction will be biased. But how about the GEs are independent? In that situation, we know the point estimator, the regression coefficient will be okay. But how about the testing? Suppose the true model is the effect is a quadratic effect, but we misspecify the model. We assume the linear effect of the exposure, and then we consider the interaction term and the main effect of the G, and then main effect of E, and also GE interactions. So here are the results. So you can see that this is just a naive regression. And then using the standard approach by fitting this model, and then we look at, we misspecify the exposure effect of E, and then we check whether the interaction term between G and E is significant. And then this is under the null. Then what that means is the beta under the null hypothesis when the interaction, there's no gene and environment interactions. And so you can see when the continuous outcome and model is fit, suppose like the Y is MPI, and then you can see that kappa error rate is off. And so because misspecification of the exposure effect. And even though the point estimators are of the regression coefficients okay, but the test is wrong. And also, if we want to look at the binary outcome, like a disease outcome, so for example, whether a child has a neurodevelopment problems, and this will be binary outcome, and then you can see that the type error rate is conservative. And so sample size does not change the conclusion. So you can see when the sample size increases, we still see the type error rate is off for both the continuous outcome and also the binary outcome, and when the exposure effect is misspecified. So how can we fix the problem? And when the exposure effect is misspecified? So a classical approach is to use the sandwich approach and to correct for the standard error of the gene and the environment interaction. And so why it is called a sandwich? And generally the form takes like sandwich form, like ABA form. And sometimes this is called robust standard error. And so this can be calculated and from many of the softwares like in R or SAS. And so let's see how that works. So this is the sandwich standard error estimate. And so you can see that when we apply to the continuous outcome, it's still off. And for binary outcome, it's roughly okay. And so even when the sample size increases, and we can see that when the sample size is moderate and the sandwich estimator, the type error rate is off. But when the sample size increases, it's become better. So why sandwich estimate does not work well and for moderate sample size? And so in the literature, there has been multiple papers suggesting using the sandwich estimator to provide a better testing. And so basically correct for the bias of model-based standard error estimator. But in the Superfund data, and so in Bangladesh data, we have 500 samples, and in the Mexico data, we only have 400 samples. And so as the sample size is not very large. And so as you saw from the previous slides, the sandwich estimator does not work well. It could result in inflated type error rate. And so that means the p-value would be wrong. Why is that the case? The reason is the sandwich estimator is biased downward, and also that's subject to additional variability. So therefore, it can lead to elevated type error rate. So how can we do better? How can we get the correct standard error estimator and make sure the type error rate is correct when we analyze the Mexico and the Bangladesh data? So this, as I mentioned, the goal here is we are interested in testing gene and the environment interactions, and then we want to control the type error rate to make the QQ plot look right, and for the gene and the environment interaction. So that is what we proposed, and we use the we call the BISC method. This is a bootstrap inference with a corrected sandwich approach. So that is shows in this red circle here. And so this is the type error rate control. So you can see the true type error rate is 0.05. And then for both the continuous outcome and the binary outcome, by using this procedure, it seemed like it worked quite well. They are close to the type error rate 0.05 level. And for both the moderate sample size and also the large sample size. The first scenario in particular is similar to the Bangladesh and also Mexico data. So this is encouraging. So now let's see how this works in analyzing the Harvard Superfund data when we do the genome-wide environment interaction association analysis. And so in this situation, the Y will be the MPI or DPI. And so for the interest of time, I'm going to show you the result of MPI. And then the exposure is the lead. And the SNP is the G, and we analyze one SNP at a time. And the confounder here include the sex, birth weight, and the first two ancestry principal component, and gestational age, mother's education, household smoke, and also child age. And so the goal is we want to look at a lead by SNP interaction. So here is the result, as you recall, when we use the naive approach and we saw that the QQ plot for the gene and the environment interaction is off, and this is a red curve here, and especially for Mexico data. And so the reason is, as I mentioned, we saw that this is due to the misspecification of the lead effect, the main effect of lead. And so when we repeat this analysis by modeling the exposure effect using the spline, and the QQ plot looks better. And then we also showed that the QQ plot for the G effect is okay. But then the question is, suppose I still want to use the linear exposure effect for lead, but now I'm going to use the Bisq method to correct for the standard error. How that work? And so to do that, in order for that Bisq method to work, and then we need to make a few assumptions. The major assumption is we need to assume the gene environment interaction. And so then here, then basically we need to assume the SNP and the following environmental effects, the lead, and also the interaction between the and also the exposure effect here, also the confounders like sex, birth weight, and the principal component, gestational age, and the mother's education, household smoke, and child age. We control for those Ws, and this part may be misspecified. And so we use a Bisq to calculate the p-value. So I want to mention here, we still model the lead effect linearly in this model, but we provide the correct p-value calculation using the Bisq procedure. So here are the results. So as you can see that this red curve, this QQ plot, and for the gene environment interaction departs very early on, and for the Mexico data. But when we apply the sandwich estimator and also the Bisq procedure, you can see that it's better. And furthermore, when we do the meta-analysis, as you can see that the red curve and QQ plot for the gene environment interaction is still off. And then because of misspecification of the exposure effect, and also the sandwich estimator seem result in the inflated taper error rate. But when one use the Bisq, and so this is the green curve, you can see it has very nice 45-degree line. And so we also look at, I want to highlight one particular gene. And so this gene is this UNCFD gene. And so this, the red, and the red is the PCI, and also the blue is MDI. And so you can see that we found a good evidence, but doesn't reach the genome-wide significance. When we analyze the two SNPs separately, there was some evidence of these two SNPs, which shows a gene environment interaction. And then later on, we decided to combine these two SNPs. And then when we combine these two SNPs, reach genome-wide significance. And so summary, when we do MDI meta-analysis, so those two SNPs we found have a moderate effect, but they did not reach genome-wide significance. And so this UNC59 gene is an interesting gene. This encodes a receptor for natrin, which may be involved in exon guidance. And since those two SNPs were not in LD, so we combined these two SNPs, and then by adding in their allele count, and then we found out the combined SNP reached the genome-wide significance when we look at for gene environment interactions. To summarize, and so in the genome-wide environment interaction studies, the environmental effect may be misspecified. When the environmental effect is misspecified, and then the gene environment interaction estimate can be biased, and also the taper error rate can be inflated or deflated. So when the sample size is moderate, like the Bangladesh study and also the Mexico studies, and the gene environment interaction using the sandwich estimator can be conservative, but the sandwich estimator work well for large sample size. And we found that when we use this BISC method, and so this is using this bootstrap corrected sandwich estimator, and then this protects the type error rate for both moderate and large sample size, even when the gene main effect of the environment is misspecified. The assumption we need here is we need the gene and the environment independent. And also the analysis of Harvard Superfund data showed that UNC at C5D is a strong candidate for the interaction with lead on the infant neurodevelopment. So I want to thank my former student Ran Sun, who took a lead in this work, and also the PI of the former Harvard Superfund Bob Wright, and also my long-term collaborator David Quishalny. So those are the references of the work I presented today. Thank you.